The Center for the Improvement of Human Functioning International presents this luncheon lecture. Today's topic, new cancer research, vitamin C and angiogenesis. Your presenter, Joe Casciari, Ph.D.
Hello, how's everybody? Can you all hear me? Okay, so today we're going to be talking about vitamin C and angiogenesis. Now, angiogenesis is a process of new blood vessel growth. It's important in wound healing, fetal development, and several other things. It also is thought to play a role in cancer in that tumors recruit new blood vessels in order to allow them to expand. And one of the interesting questions of cancer research is whether or not we could stop tumor growth by interfering with this angiogenesis process. What I'm going to be talking about today is going to focus on some work that our research group, particularly Dr. Nina Mikarova, who is over there, has been doing with vitamins, looking at vitamin C and its effects on the angiogenic process. I'll divide the talk into 4 parts. We'll have a first quick little introduction to cancer. I'll discuss a little sketch of the biology of angiogenesis, and then we'll talk about vitamin C. I'll review a little of our vitamin C and cancer work, and then we'll get specifically into the new data that Dr. Mikarova has been working on. So cancer cells are descendants of normal cells that, first of all, have lost their normal function, and secondly, have lost their growth inhibition. And this is typically described in terms of oncogenes and proto-oncogenes, where genes that are normally turned off in cells become turned on, or vice versa, and this creates cancer cells which are growing in an unregulated fashion. Now, initially, cancer cells will form small nodules which are undetectable. They're maybe a millimeter or 2 in diameter. And the growth of these left to their own devices will be limited by diffusion process. In other words, it takes a certain amount of effort for nutrients to work their way all the way to the center of a tumor nodule, and it takes a certain effort for metabolic waste products like lactic acid to work their way out. So even with a small microscopic tumor, you create a situation where the cells at the center of the tumor are becoming necrotic, and other cells are kind of going into a slow quiescent state. So this limitation in diffusion in turn limits the growth of the nodule to only a couple of millimeters in diameter. And so it's thought that in order for tumors to become what they are, they need to recruit new blood vessels to grow towards them. Tumor cells do this, and we'll go into the details of this a little bit, but tumor cells do this by releasing chemicals called cytokines, which communicate with other cells. In particular, in this case, they stimulate endothelial, that is blood vessel cell growth, towards the tumor. Once this happens, the tumors have a fresh supply of nutrients and are able to expand. The other important thing that they're able to do is they will actually get into the bloodstream through this new blood supply and establish colonies in remote sites, and this is a process referred to as metastasis. So you often, by the time you've detected the cancer, you often have a large primary tumor, and you may also have smaller metastatic colonies. The typical protocol for this in standard care is to remove the primary tumor through surgery and then try to use chemotherapy or radiation or something like that to control the metastases. Now, there's a phenomenon I'll discuss in a little bit where removing the primary tumor actually causes the metastatic colonies to grow faster. And this is involved with angiostatin and endostatin, and we'll discuss this in a little while. Now, one of the problems with treating cancer cells with chemotherapy is that there are subpopulations of resistant cells, and I've tried to depict those in red in this little schematic diagram. What happens is when you treat a cell with a tumor with chemotherapy, the sensitive cells are killed off, but a handful of resistant cells survive, so that when they repopulate the tumor, the entire tumor is now drug resistant. This is why if you know someone, say, who's was treated with adriamycin and they had a remission, but then the tumor comes back, they can't treat it with adriamycin again because the new tumor will be resistant to adriamycin. It's the same natural selection process which guides drug resistance of bacteria. Tumors eventually kill the patient by invading nearby tissue, causing a wasting process called cachexia and weakening the body's defenses against other infections. Obviously, cancer has been very difficult to treat because the patient is dealing with not only side effects of the tumor itself, but also side effects of the chemotherapeutic regimen they might be on. You have some cells that are resistant to the treatments. And when I say here that the body reaction favors the tumor, meaning that tumors are inducing new blood vessel growth and they're evading the immune system so that they're able to spread. Now, some of the kind of grand scheme of things that we've been working on at the center and other places is to find treatments that work with the body, not against it. In other words, rather than using drugs that harm the body but hopefully harm the tumor more, we're looking for ideas that actually help the body and improve the patient's well-being. So this has led us to nutritional support and looking into nutrient-based therapies that would improve the patient's health while they're on the treatment. One of the ones we will talk, obviously the one we're going to talk about today, is finding efforts to inhibit this angiogenesis process, this new blood vessel growth to the tumor. I mentioned reverse pathogenic processes. This is pay attention for future luncheon lectures. If we can ever get Dr. Shalong Meng to give a talk, he will talk all about this for you. He's hiding back there now. So let's talk a little bit about angiogenesis and some of the details about how it works. One of the key molecules produced by tumors which stimulate white blood, stimulate endothelial cells is something called vascular endothelial growth factor. This is a key cytokine, and both mature endothelial cells and endothelial stem cells respond to this. In my looking this over, it seems like the issues are still being looked into as far as which response is more important and what happens when, but you have both a stimulation of the mature endothelial cells in the area of the tumor and a recruitment of stem cells from the bone marrow that all result from tumor production of this VEGF cytokine. Now, a little bit about the characteristics of the endothelial cell response. They produce what are called proteinases, which will digest the extracellular matrix. This will be a key step in allowing the new blood vessel to branch forward. They also produce something called, they contain something called integrins, which are kind of grappling hooks for these cells. So this is all part of the migration process of building a new blood vessel. Now, these won't be on the exam, so don't worry if these terms go by you. But another key constituent is nitric oxide, which endothelial cells produce, which regulates the angiogenesis process. And I would just want you to keep these a little bit in mind because they're potential targets for inhibiting angiogenesis. This is just an example on a chick embryo of what's called the allantoic membrane. And normally this is well vascularized as the baby chick is growing. But if you place a cell medium that has been conditioned by tumor cells, in other words, tumor produced cytokines, notice how there's a dramatic change in the vascular pattern and it's all shooting right towards the area where the tumor would be. I also want to point out that there's something of a positive feedback effect involved here. And a lot of what's happening in cancer is the feedback between tumor cells and normal cells. One obvious thing is that the tumor cells are stimulating endothelial cells to form new blood vessels, and this in turn increases the tumor cell number, thereby increasing the stimulatory signal. Also, there's an interplay between white blood cells and tumor cells, which sometimes favors the tumor cell, because white blood cells can also stimulate endothelial cells and so forth. Now remember I mentioned earlier that sometimes you remove a primary tumor surgically and the metastases grow more quickly. This is due to materials called angiostatin and endostatin, which are produced as a result of the tumor. And now what makes this a little interesting is that these actually will inhibit angiogenesis. So it seems like the tumor is working at cross purposes in that it's producing angiogenesis promoters and producing angiogenesis inhibitors. The difference is that the angiogenesis promoters tend to act locally, whereas the angiogenesis inhibitors tend to act more systemically. This means that at the site of the primary tumor, your concentration of your inhibitor is much of your angiogenesis promoter is much higher than that of the inhibitor, so you get angiogenesis at the site of the tumor. However, at the metastatic nodule, it's the other way around. So the metastatic nodules are just kind of hanging out and not growing too quickly. However, when you surgically remove the primary tumor, you cut off the supply of the angiostatin and endostatin, and this allows the metastases to kind of grow into large sizes. So what all of this kind of studying the mechanisms of angiogenesis does is give us potential targets. One would be interfering with the action of VEGF, the main cytokine that promotes angiogenesis. The second would be interfering with the endothelial response to that, meaning endothelial cell proliferation, endothelial cell production of digestive proteins, and endothelial migration, particularly with the integrins. And so these are all potential targets of angiogenesis inhibitors and potential things that can be tested experimentally in the laboratory. There does happen to be, according to my searching, one actually FDA-approved angiogenesis inhibitor. It's called Avastin, and it is an antibody to VEGF, so it's trying to block that particular cytokine. In I think it was a phase 3 trial of colorectal cancer, it increased survival time by 5 months. And it does have some side effects. So the search kind of continues for other angiogenesis inhibitors. Yes, thalidomide is the one that was causing birth defects about 30 or 40 years ago, but that apparently has angiogenic properties, so the FDA has actually approved it for study in cancer. Also, there's a product of shark cartilage, and as I mentioned, angiostatin and endostatin. There are also some natural products that are thought to inhibit angiogenesis, and of course the one we're going to be focusing on today is vitamin C. So let's go forward. First, what I want to do is review some of our history and our work with vitamin C in cancer, and then I'll get specifically into the effects of vitamin C on angiogenesis. First of all, vitamin C is primarily known as a water-soluble antioxidant. It's important in protecting neutrophils and other white blood cells as they do their job of eliminating toxins and infections from our system. But it can also, at high concentrations, and I'll get into this in a little bit, it can have a pro-oxidant effect. It has several biological functions. It's essential for proper functioning of the cell-mediated immune system. It is important in the production of collagen, which strengthens the extracellular matrix material, and carnitine, which does a similar thing. Now, a lot of the initial information on vitamin C was with oral administration. One of the big breakthroughs as far as cancer is concerned is the idea of introducing vitamin C intravenously. So I'll use the term IVC a lot as we go along, and that means we're talking about slow intravenous infusions of vitamin C. And what this does is give you, if you've never worked with this before, you're kind of shocked to hear that 10 grams or 100 grams of vitamin C can be given to people, since the quoted daily recommended allowances are in the milligrams, but we'll get into the rationale for that in a little bit. At the center, we've been using vitamin C for decades. Our clinical experience with this is, first of all, that we find that cancer patients seem to be particularly depleted of vitamin C. They have very low levels of vitamin C in their plasma. So there's a vitamin C sink. It's thought that the tumor might be soaking up the vitamin C, or just that for whatever reasons, cancer patients are often low in vitamin C. And so the simple act of replenishing their vitamin C stores makes them feel better, for the reason regaining nutrition would make anyone feel better. Secondly, we've had several cases where by administering intravenous amounts of 10 grams to 100 grams, we've been able to reverse cancer or produce positive outcomes in patients who had cancer. This has both been in cases where it's been combined with other treatments and some cases where it was just administered alone. We actually funded a phase 1 clinical study several years ago, and the results of that are out, just to ensure that vitamin C could be administered safely at these levels, and it can. I'll discuss some of the details of that. Phase 2 studies are underway at several sites around the country as interest in this has picked up. The non-trivial question is, why does vitamin C do what it does? The original mechanism proposed by Linus Pauling way back in the day was that it would wall in tumors by strengthening the extracellular matrix. It's also thought to, obviously, since it supports the immune system, and particularly the sections of the immune system that are responsible for attacking cancer cells, it's thought that it certainly can't hurt the body's immune response against cancer. Vitamin C is also actually at high concentrations. Vitamin C, and this is the pro-oxidant function I talked about, it can lead to hydrogen peroxide production. And while hydrogen peroxide is an oxidant, it is kind of selectively toxic to tumor cells because tumor cells lack an enzyme called catalase, which would normally neutralize it. And the relatively recent discovery that Dr. Mikarova has been working on is that vitamin C can also inhibit angiogenesis. And we're still, interested in which of these mechanisms are really the important ones. Is all of them put together or what have you? The first thing we really took off and ran with was the selective cytotoxicity due to the hydrogen peroxide generation. And let me show you. First of all, the chemistry behind that is right here. Ascorbic acid is reduced to dehydroascorbate, and this causes some particularly nasty oxidants to be converted to hydrogen peroxide, which itself is capable of damaging cells, but under normal conditions, normal cells can neutralize it with catalase. So this leads to a potential selective toxicity towards tumor cells. Now, at first, no one was particularly interested in this because the concentrations needed for this effect were considered too high to be achieved physiologically. However, this was before it had been demonstrated that intravenous administration could be safe. So a lot of our initial work, and this is a lot of the work that Neil Riordan in particular spearheaded, was comparing the concentrations of ascorbate needed to kill cancer cells to the concentrations that we could achieve with intravenous infusions. And so that's what this ugly graph is symbolizing. This is a measure of the vitamin C level as a function of time during infusions. And it gets up to, I'm going to throw a couple of different units with you. These are milligrams per deciliter. It gets up to what's called the millimolar level, which is a relatively high level, much higher, a couple of factors of 10 higher than you could get with oral administration of the drug. And then we were able to compare this with, compare these levels achieved through IV administration with levels that we'd found in the laboratory killed cancer cells, and we got a match, meaning that we are able by intravenous infusion to attain vitamin C levels in the blood sufficient to create this toxic effect in tumor cells. We also, by the way, did some experiments with vitamin C combined with chemotherapy and found at sufficient concentrations it had an additive effect. Also, I want to mention alpha-lipoic acid. is something we found enhances the efficacy of vitamin C. So this led to the idea of intravenous vitamin C with other supplements such as lipoic acid could actually have a cytotoxic effect towards tumors. And this is really interesting because vitamin C actually accumulates in tumors. So you have something that's toxic towards tumor cells and collects in tumor cells, so that was considered a good sign. Apologies to the animal lovers, but I've done some work in the past with guinea pigs and showing that when you look at guinea pig tumors, whether or not they've been treated with vitamin C, we were actually able to measure the intra-tumor vitamin C levels, and there was a strong inverse correlation with tumor size, meaning that in treated guinea pigs, we were attaining high intratumor ascorbate concentrations, and these were directly correlated with very low tumor sizes relative to controls. So this was another area of interest to us. So all of this is motivating the use of intravenous vitamin C. We did, as I say, do a phase 1 trial with intravenous vitamin C, going from 10 to, I believe, up to 60 grams per day. And what we found was that, first of all, there was concern as to whether vitamin C would, those high concentrations in the plasma would affect the white blood cells. Well, white blood cell counts for these patients remained stable. Also, blood chemistry parameters related to renal function remained stable also. There were some concerns since vitamin C is water soluble, it would be filtered out by the kidneys, and there were some concerns as to what would happen to the kidneys. Well, it did not affect renal function in these patients. There was actually one case out of the 24 or so where we actually had the patient, these are all patients who were pretty much at death's door, had months to live, but there was one patient who actually had a stabilization of their disease and was able to go on for a couple of years with the treatment. Some of the adverse events we found, one person did get a kidney stone, and this was a person who had a history of them. So I put that there as kind of a warning, although I think that if you don't have a history with kidney stones, you're not going to have a problem with them. That's kind of an urban legend that's been going around with vitamin C, that it would cause kidney stones, and the really evidence for that isn't there. Secondly, we found that edema and low potassium levels were found. That's a relatively minor effect compared to, say, having cancer, but it's something I want to inform you of. We've developed what's now called the Riordan protocol for intravenous ascorbate. It's available upon request for physicians anywhere in the country or the world who would like to know, well, how do you actually administer intravenous vitamin C to people? The key things are, we recommend a slow drip with a dilute solution, obviously, to prevent hyperosmotic conditions from being a problem. I mean, when you expose cells to a high concentration of any ionic salt or substance, they are in danger. And so you give a dilution and a slow drip. What you want to do is start slowly and then ramp up the dose gradually. And at the same time, we think a key to this is monitoring the blood vitamin C levels during the infusion. And actually what we can do is correlate the peak concentration to what we think we need based on some of the data we have in cells and guinea pigs, what have you. And so this gives a target range of what kind of doses you'd like to give. And we're actually finding that you don't need to climb up to 100 grams or so to do the trick. That, steady 15 to 30 gram infusions can do the job. Now, as I mentioned, we're not sure whether the benefits of vitamin C are strictly due to this hydrogen peroxide generating mechanism we've discussed. In fact, a lot of us tend to suspect that its overall biological effects might be more important. And Dr. Mikarova, who is right over there, yeah, let's make her feel worse. No good deed goes unpunished. She began studying the effects of vitamin C on angiogenesis. Now, this is actually an area of research we've been interested in for some time. I had been doing some work with it back when I was living and working here. Dr. Shalong has done some work on angiogenesis, particularly plant extracts that inhibit angiogenesis and so forth. And so Dr. Mikarova decided to look in vitamin C in this regard. In her experiments, she used both mature endothelial cells and endothelial progenitor cells. She actually developed a technique to isolate endothelial progenitor cells from what we call peripheral blood, meaning blood you would draw out of someone's arm or something. So what we call the HUVEC, which is short for human umbilical vessel endothelial cells, I think. These are mature cells that you can grow in the laboratory for a certain length of time if you expose them to heparin and other growth factors and give them lots of TLC. And in particular, I'll show you some pictures of this. They can, in the Petri dish, be made to form a network of blood vessel-like tubules, which gives you actually a visualization of the angiogenesis process. The progenitor cells can also do this, tubule formation. So we have both in our laboratory, both mature and stem-based endothelial cells that we can conduct experiments on. Now, one of the things you have to do, and again, this won't be on the exam either, is you have to establish that the cells you're working with are really the type of cells you think they are. And cells have characteristic markers on their surface which identifies them as a certain cell type. And these markers can be detected with fluorescently tagged antibodies. And we have a flow cytometer, and we have fluorescent microscopes and fluorescent plate readers. And between all these things, we can basically tag these cell markers with fluorescent tags and verify the presence or absence of certain markers. And this allows us to match them to their phenotypic expression to verify that this is what HUVEC cells should have, this is what the epithelial progenitor cells should have. So Dr. Mikarova is able to verify that the cells she's working with are indeed endothelial cells. And we investigated angiogenesis by several different assays. Let me show you first of all. This is how endothelial cells look when they're just growing on a Petri dish. When you let them keep growing, they get crowded together and they form this nice cobblestone formation. So obviously the first thing we can do right off the bat is we can determine whether or not whatever we're doing to treat the endothelial cells will affect the endothelial cell growth. Now, on a substance called Matrigel. Now, Matrigel is a sort of artificial extracellular matrix material that you can buy. It at refrigerator temperatures, it's a liquid. When you let it harden at room temperature, it forms a gel. And then you put your endothelial cells on top of this gel, and they do this. They rearrange themselves and form these tubule networks. Now, what Nina has been able to do is to analyze these quantitatively by counting the number of what we call closed loops. For example, this is a network of tubules that forms a closed circle. So in our analysis, we would be able to count that as one, and you can count how many of them there are in a given area on your Petri dish, and that gives you a measure of the extent of tubule formation. Which, of course, since this essentially is the migration and formation process of angiogenesis, that's giving you a measure of whether or not angiogenesis can occur. There are other types of assays we'll show you. One is an aorta ring assay. We look at the ability of cells to migrate under certain conditions, and we look at the ability of cells to grow into matrigel from a surrounding area of tissue. Also, Dr. Mikarova looked at endothelial cells to see how their functioning is affected by vitamin C. For instance, how does vitamin C affect their proliferation, their production of ATP, in other words, their energy status. And how does it affect nitric oxide production, which, as I showed you earlier, is an important mediator in angiogenesis. So now we can go through some of the data. First of all, here are some pictures of tubule formation for both the progenitor cells and for the mature endothelial cells. And you can see here that with the controls, you have a pretty involved network, more so with the mature cells, but in both cases, a pretty involved network of newly formed tubules. You can see that this is impaired to at least some degree in the presence of vitamin C. And when I say in the presence of vitamin C, I'm referring to concentrations on the millimolar level. These are concentrations that would be relevant in the case of intravenous infusion of, say, 10 to 30 gram levels, but they're not the kind of thing you have just occurring in your body normally if you're taking vitamin C tablets. Graphing the percent of closed loops compared to the control, you can see that this inhibition is concentration dependent. The more vitamin C you have at very high concentrations, the less angiogenesis is formed. So vitamin C tends to be inhibiting the new vessel growth and kind of promoting a stability of the tissue, if you want to think of it that way. Another model we have. Okay, first of all, I cheated. This picture is not really a zoom up of that, but I had a low mag and a high mag picture, so I pretended a little bit. But what happens is you have a section of aorta, and under the right conditions, endothelial cells will branch off of that in an angiogenic process. And you can measure the extent of this branching and compare controls to some that were given. I think this was 3 millimolar of ascorbate, and there's a large inhibitory effect with time. Now, there's also a Matrigel plug assay where you can insert, remember I told you how you could coat a Petri dish with Matrigel and the endothelial cells would arrange themselves and form these tubules. Well, you could also put this into a section of tissue, like under the skin, and observe vessel growth into the Matrigel, which, if angiogenesis is occurring normally, endothelial cells would be encouraged to grow into the Matrigel. So here's a histological section. You know, obviously out here is just the plain old Matrigel, but in here are some blood vessels that are growing into it. So again, you could examine slices of this and look at the extent of blood vessel formation. And you can compare them between untreated hosts and hosts with 400 milligrams per kilogram of ascorbate. And that, see, multiplied by 70, that's about equivalent to a 25 to 30 gram dose in your run-of-the-mill adult. And this shows that in 3 different replicate experiments of this process, we see anywhere from a 50 to a 70% decrease in the extent of blood vessel formation. Another assay Dr. Mikarova conducted was what's called the migration assay, or the wound healing assay. This is real simple. You get a Petri dish full of endothelial cells, and then you come along with a plastic scraper and you scrape the middle. You make a scrape in the middle. So now, microscopic overview, you have a pretty big gap between the 2 sections of endothelial cells. Now, if the endothelial cells are growing and proliferating and stimulated to do such, they will grow in and try to close this gap. And so what we could measure is, you can actually measure the gap thickness, if you will, or and the higher the gap thickness, the less healing, quote unquote, you have. And so the gap closes much more quickly in control cells than when you have cells in presence of high doses of vitamin C. Okay, first of all, you get to the end of a 45-slide presentation, you start getting goofy. So this is my daughter, Anna, on the keyboard, and this is my son, William, playing with the cars. And this is, you're not supposed to distract the audience from the data, so I apologize, but I couldn't resist. Anyhow, what happened here is this is a situation where Dr. Mikarova looked at the actual cell performance of endothelial cells. First of all, she found that there was only a slight decrease in ATP production, and there was no effect on cell viability. So the high doses of vitamin C didn't actually harm the endothelial cells or shut them down metabolically. It did, though, decrease expression of cahedrin, which is one of those grapple hook molecules that help the endothelial cells migrate and latch on, and it also inhibited nitric oxide production. So here you see the grappling hooks are reduced by about 40% or so in a dose-dependent fashion, and less dramatic but still noticeable, there's a reduction in nitric oxide production, which, as I said, they will produce nitric oxide when they're undergoing angiogenesis. So at the cell level, we're seeing vitamin C affect endothelial cells in such a way that they're less likely to form new blood vessels, but they're not any less viable or metabolically active. So let me draw some conclusions from this. First of all, several of the endothelial cell functions that are key to angiogenesis, in other words, several of the targets we looked at, are affected by vitamin C. Endothelial cells are less likely to migrate to produce nitric oxide. They will have a more difficult time forming the tubules and penetrating through the extracellular matrix under high concentrations of vitamin C. And when I say high concentrations, I'm referring specifically to concentrations that we think are clinically relevant in the case of high-dose intravenous administration. And this is potentially exciting because angiogenesis is really one of the keys to tumor growth and tumor metastasis. And if you can really inhibit new blood vessel formation in tumors, then you could inhibit tumor growth and perhaps tumor metastasis also. So in a sense, while we're learning more about vitamin C and angiogenesis, this is shedding us a new interpretation on any of the clinical results we've been getting with intravenous vitamin C. Now, in addition to the hydrogen peroxide mechanism, which I've discussed quite a bit, we're really looking strongly at vitamin C's effect on biological response, particularly on angiogenesis, and it goes into this whole paradigm of working with the body instead of against it, because vitamin C can also improve well-being, boost the immune system, and so forth. So that basically is what I have for you. And some of this work, Dr. Mikarova's work has recently been published in the Journal of Translational Medicine, and we could certainly get you that paper by request if you need it. I wanted to point out a few other things. The classic papers are Dvorak, Tumors, Wounds That Don't Heal, and just about any review article by Dr. Judah Folkman, who recently passed away, I hear, but he was kind of the discoverer of tumor angiogenesis. And he also was the pioneer in a lot of the work with angiostatin and endostatin. If you want to find more information on vitamin C, actually I had written a protocol summary and literature review a couple of years ago, but since then the doctors at the center have written a much better one from a clinical perspective. So, yeah, I'd be glad to give you the one I did, but you can also get, by request from the center, the one they make available to physicians. It also would have any of the early references to any of the preclinical work we've done with vitamin C or anyone else has done. And I want to mention Dr. Mark Levine at the NIH. He had gotten interested in this. He had known Dr. Riordan, and he's done a lot of work with, first of all, establishing what normal concentration ranges of vitamin C are, then looking at the difference between intravenous and oral administration, and obviously when it's someone from the NIH and they're publishing in Proceedings of the National PNAC, that gets people excited. So he's been getting a lot of publicity for this work too, and that's been adding to the interest of vitamin C. And then last, this is Dr. Mikarova's recently published manuscript, which contains some of the data that I've shown you in the talk. And so when you prepare a lecture, you have 42 slides, and you think, oh, geez, I'm going to have them here all day. And then you go through it, and it feels like you race through it. So we actually have some extra time for questions then, I believe. So thank you very much.
Just raise your hand and we can get your questions answered.
I'm on warfarin, and I was told at some point in time that I couldn't take vitamin C. Is that true?
Okay, say again which drug you're on?
Warfarin for blood clots.
Okay, yeah, I don't know the answer to that. Dr. Ron does, though.
No, you can go ahead and take vitamin C with warfarin. I just tell people to start low and to monitor your pro time, but by all means you can use vitamin C. It just becomes a question of dose.
You don't want to take K though.
Yeah.
What would be a good starting dose for someone just wanting to start?
1,000 milligrams.
A gram.
I get one shot a month. It's called Sandostatin, and I've been doing everything I can on my own by going to the health food stores and trying to handle it myself, as well as exercise, proper diet, and the surgeon and the cancer doctor are ecstatic with my well-being. They said that my surgeon says he's never seen anybody get better without further treatment or further surgery. So I'm a believer then in this natural way of trying to get out of the woods. So, yeah, I'm interested in how much vitamin C can I get when I'm on the Sandostatin shot once a month.
I don't know precisely about that specific medicine, but once again, I think you'd have to sit down with one of us and we'd have to look that up. And we have an oncologist on staff that would kind of help us work out the details of that issue.
Thank you very much for your lecture. As a medical student in town, we don't get exposed to all of this, and I appreciate your time.
Thanks.
I was wondering, since not all tumors are created equal, if certain tumors in the literature respond better to vitamin C than others, if these studies are mostly adenocarcinomas, glandular-forming tumors, common to breast cancer and colon cancer.
The ones off the top of my head I remember as having the best luck with, now obviously the preclinical research was general with a lot of different tumor cell lines, but clinically metastatic renal and breast cancer, we've had positive results with those. I think not so much with colorectal, although I could be wrong on that. So yeah, you're right. I think I would say those two are the ones that I remember us having the most dramatic successes are, the metastatic renal and the breast and maybe the ovarian. I'm looking at Dr. J and Dr. Ron to see if I'm wrong, but that's what I recall.
It's not just the tumor type, because we have a 6-year survivor of colon cancer that was stage 4, but she did a combination of IV vitamin C and chemotherapy, and it was a very effective combination. So it's not what tumor cell you have, but it's who you are and what you're willing to do to get better, because very often getting over cancer is several interventions. Vitamin C being one, dietary factors, lifestyle. There's just a lot of things. Your belief in getting well, I think, is a very important part of this process. So the tumor types certainly do have a role to play, but who you are is more important than who the cancer is in my books.
We've also had a patient with pancreatic cancer, head of the pancreas, which normal lifespan, I mean, after you've been diagnosed is about 4 months, 5 months. It's been alive for over 3 years, which is amazing when you consider this stuff. But we don't have a tumor registry because we're not a cancer treating center. We're a center who will treat patients with cancer if they come in and want to be treated. See, we're not actively, doing that. But so as far as the statistics that, you were asking about, we can go and sort of look them up, but we don't have all that data. But renal cell carcinoma is in breast is good. We had 7 years, 8 years people with this stuff. Metastasis, renal cell metastasis to the lung and liver completely cleared up. Person's still alive. So those are like Joe said, those are the two types.
You've been talking about solid tumors. What about, say, leukemia? Is it effective on that?
I would think so. I would think naively that it would benefit because then you're right in the bloodstream where the intravenous vitamin C is working. So I would think I would suspect no more or less with leukemia than other types.
With alpha-lipoic acid, really.
Yeah.
It's recently published a paper where the alpha-lipoic acid actually killed the leukemic cells. Dr. Nina Marikova again was doing this, and it didn't do anything to the normal cells. So it's we're still investigating that, but yeah, we've had leukemic patients here treated, but again, we don't have statistics and data. We treat patients, we don't treat the data.
In your research, you discovered that people who had cancer had a lower level of vitamin C in their bloodstream. In a preventive measure, what level of vitamin C should you be maintaining?
I guess the answer to that in part is that depends on you, and let me go into a little bit about vitamin C requirements and things like this because there are several levels of thought on that. The minimum daily requirement obviously is the lowest level needed to prevent prisoners, as it was tested on, from dropping dead of scurvy. It's thought that, from Dr. Levine's work, it's thought that a healthy adult who is fully nutritional, supported with all of their other nutrients, could do well with, say, 100 milligrams a day. Now, on the other hand, if you make an argument by analogy with how much vitamin C do other primates consume in their diet, and how much vitamin C is manufactured by animals that can manufacture their own vitamin C, and scale that up kilogram-wise to human beings, that gets you on the order of 1 to 5 grams per day being a normal dose. Vitamin C is depleted under situations of stress, so if you're, Dr. Riordan used to always give me the example of nurses working an 8-hour shift, their vitamin C plasma will be 0 at the end of this shift. And so our approach when we're at the center has always been to measure it, see what your plasma level is, and keep boosting your dose until you have a healthy plasma level. So I personally, if you're just a healthy person, you want to know how much vitamin C to take, I kind of think a gram is reasonable, and Dr. Jackson could help with that.
Paulson calculated about 2,000 milligrams a day.
Yeah.
And also, a nice way to check your urine to see how you're doing, Joe said stress will wipe it out. And the two most common symptoms of low vitamin C is irritability and fatigue. My wife says I need a rolling IVC every time because I'm tired and cranky all the time. But you can get a bottle of C-stick, VitaCheck C-stick up here, and measure your urine every morning. And if your urine on that strip is not in the yellow range, then your body is using up every bit of vitamin C you have, and you're not peeing it out. Now, if you got your tissue saturated, being water soluble, you would pee it out. So if it's green, that means you need more vitamin C. So that's a quick, easy way of seeing how well you're doing. Just dip the strip into urine, 30 seconds, read it, compare it to the color chart. It's a quick and easy way. It's a screening test. It's not a definitive test. You can buy them upstairs, by the way. Very good product.
I'm picturing Dr. Jackson going home like Archie Bunker, but instead of Edith getting him a beer, Edith's getting him a vitamin C. He still comes home all aggravated.
I would like to mention I've used those Vita C check strips on several occasions, and during the day it's very beneficial to measure because during the day when all the stress hits, it does deplete. And it is a very useful tool to use, and as Dr. Jackson mentioned, we do have those upstairs in the Gift of Health. We do have a few minutes for another question or two. Just raise your hand and we'll be around with the microphone.
I like to tell people that when my grandchildren were born, my son and my daughter both having previous cancer, we harvested the stem cells from the umbilical cords, and you can do that now, and they're in a vault out in Arizona someplace.
Mm-hmm. Yeah, and we're doing a lot of research with stem cells, and in particular how progenitor cells could interact with cancer cells. I discussed the cancer cells and the endothelial cells communicating through chemical cytokine signals and so forth. Well, we're researching how that could work with, say, how, for instance, progenitor cells could affect the growth and development of cancer cells, and Dr. Shalong has a lot of work on that.
The preceding program was presented by the Center for the Improvement of Human Functioning International in the Bright Spot for Health Lunch and Lecture Series. To inquire about additional health-related information available on DVD, audio CD, VHS, or audio cassette, simply call 316-682-3100 or drop by 3100 North Hillside in Wichita, Kansas. To discover more about the center and what we have to offer, be sure and visit us on the web at www.brightspot.org.